Alzheimer's disease, and as Dr. Albers mentioned before, Alzheimer's disease is a terrible disease. It's a progressive disorder that actually leads to memory loss and also the loss of the thinking skills, leaving us to the inability to complete even the simplest task. As mentioned before, more than 6 million Americans are actually living with this disease. It's also one of the most expensive disorders out there, with estimated costs over $300 billion. Despite not having a cure, there is hope. And something that I'm very excited about it is like right now we have the opportunity to detect the Alzheimer's disease more than 20 years before people develop symptoms, which has opened an opportunity to delay clinical onset and also thinking about ways to prevent the disease altogether. Even more exciting, we have now the opportunity to actually think about treatments that can modify the disease progression, and we have treatments, novel treatments to address the symptoms of the disease. We also have, as was mentioned before, the regulatory pathway or approval for treatments in asymptomatic stages of the disease. However, as you may be aware of this, historically, most of the treatments for Alzheimer's disease have focused on the reduction of pathology of the disease. This is amyloid and tau pathology. Interestingly, and as Dr. Albers mentioned, there are many individuals that have amyloid pathology or tau pathology that don't develop cognitive impairment or dementia. So what if we can have alternative treatments that are not only based on the pathology burden? Sporadic Alzheimer's disease or late onset Alzheimer's disease is the most common form of the disease. And this one is diagnosed generally in individuals after the age of 65. Our approach has been to study families with autosomal dominant Alzheimer's disease that usually leads to earlier onset. This has given us the opportunity to study individuals that are destined to develop the disease with near 100% certainty. For the past 20 years, since I was in college, I had actually the opportunity to study individuals for the large extended family with a single genetic mutation, what we call the PISA mutation. In Colombia, we have identified so far more than 6,000 individuals carry this mutation, who belong to this family, and about 1,200 who carry this mutation. Those who carry the mutation, they usually develop mild cognitive impairment at the age of 44 and dementia at the age of 49. In 2014, when I joined the Faculty of Mass General, I launched the Colombia-Boston study, what we now call Reelin-Colbus, and since then, we have been bringing members of these families to Boston for biomarker and other genetic examinations. In 2017, we launched what we call the Colbus Extreme Project in an effort to characterize those individuals who belong to these families but who remain cognitively unimpaired until older ages, and who have resisted the pathology of the disease until these ages, in an effort to find genetic protective factors that can explain resistance or resilience against dementia. In 2019, we published the first case from one of these individuals. This is the case of Doña Aliria Piedrahita. She belonged to this family, so based on her genetics, she was destined to develop dementia in midlife, but only developed mild cognitive impairment in her 70s, which is already showing extreme protection against dementia. She belonged to the Colbus project, so she actually came to Boston, and when we actually look at her brain, we saw that she had really high levels of amyloid compared to a typical patient from these families, but she had very limited levels of tau pathology and neurodegeneration. When we actually look at her genetics, we found that she was homozygous for a rare mutation in the ApoE gene. This is the mutation that we now know that is the Christchurch mutation. In our first analysis, we learned that the Christchurch mutation was important for the binding between ApoE and a type of sugars that are the HSPGs. And as you may know, so the HSPGs have been implicated in Alzheimer's disease at different stages of the process. They have been implicated in amyloid formation, amyloid aggregation, neuroinflammation, tau uptake, and propagation. So our first in vitro studies actually show that the Christchurch mutation impair this binding between ApoE and heparin, and it actually shows the weakest binding to heparin, even weaker than the ApoE2 that is considered to be the most protective isoform. Then we asked the question if we could elicit the protective effects of Christchurch using an antibody, and as a proof of principle, we developed an antibody against ApoE that was actually effectively able to inhibit this binding to heparin, suggesting potential for therapeutic intervention. Then we designed and characterized a library of antibodies that bind to this heparin binding region of ApoE, and identify a top candidate that is 7C11 that shows the highest binding affinity to ApoE. Then in vitro experiments show that this antibody was able to reduce the toxicity of ApoE, and also in a mouse model, we show that this antibody was able to reduce the tau pathology, showing some preclinical efficacy of this candidate antibody. Then continue with our Colbus efforts characterizing these escapees from the Colombian families. Just a few weeks ago, and you may have seen this in the news, we reported the second case with extreme protection against Alzheimer's disease. This was an individual also from these families, so based on his genetics, he was destined to develop dementia in the midlife. He only developed mild cognitive impairment again much later in his 70s, and he was part of Colbus. He came to Boston. We also observed that in his imaging, he had really high levels of amyloid. He had really elevated levels of tau pathology in several regions of the brain. But super interesting, there was one region of the brain that was the entorhinal cortex that was very limited in terms of tau pathology. We look at the genetics. He was not a carrier of the Christchurch mutation, that we discovered a novel mutation in the Reelin gene that we actually call the Reelin-Colbus mutation. What is super interesting about this finding is that ApoE and Reelin, they actually share and compete for similar receptors. They are in the same pathway. We also know, and we learned this before, Reelin signaling has been implicated in Alzheimer's disease, including tau pathology and neuronal loss. And further investigations that we conducted also led us to learn that the mutation was located near the HSPG binding areas of Reelin, which is also important and shows us that these interactions have a really critical role for the signaling on Reelin. And you may remember that I also mentioned HSPG in the context of ApoE in the Christchurch mutation. So it's very interesting to see all these key players, the common key players in the protection for Alzheimer's disease. So we conducted several experiments to study this gain of function of the mutation. We found that the mutation enhances the DAD1 signaling, that is the binding partner for Reelin. It also enhances the binding affinity to heparin. And in a mouse model that expresses tau pathology, we were able to show that the mutation reduced the pathology and also even rescue motor function. To summarize, these two extraordinary cases, the two extraordinary patients, led us to the discovery of two mutations that confer extreme protection against Alzheimer's disease via a common pathway that involves ApoE and Reelin. They also showed us that it's possible to delay the onset of Alzheimer's disease, and just imagine that we can actually delay the onset by 20 years, 30 years. For some people, for most people, that would actually mean that they won't have dementia in their lifetime. So the first mutation is Christchurch. We have an antibody. We show preclinical efficacy, and there is now a startup company that is currently developing therapy around this antibody. The second mutation that I mentioned was the Reelin-Colbus. We demonstrated a mechanism of protection. We are currently seeking partners to advance these modalities of treatments, especially the gene therapy. And as a future steps, we are very interested in seeking funding to investigate other cases that we have identified that are resistant against Alzheimer's disease, with the hope that we can discover unique protective mutations, find other novel therapeutic targets and mechanisms. And finally, we are interested in extend our program to other types of dementia, including frontotemporal dementia and vascular dementia. And with all of that, I thank you so much for your attention, and I'm happy to answer any questions.